Next, I am honored to introduce you to Dr. Stuart Kauffman, who is an emeritus professor in the biochemistry at the University of Pennsylvania and also an affiliate professor of ISB. He is well known for his work in many areas, including theoretical biology, and I know Ilya loves this area. You can talk for hours with Ilya about theory and math in biology. So thank you for joining us, Stu. We are looking forward to your presentation.
Does that work now? I am truly deeply honored to be here. I have only one slide. Can we please put up a slide, one of the beautiful slides of Ilya that we had before, a real picture of Ilya Smulovich. And otherwise make this, huh? What do I do? I don't think it's in this set. Do we have a picture of Ilya? That'll do it. I so loved this guy. I think I loved him the day I met him, something like 2000, 2001. I loved Ilya before I loved him now. So I'm going to do 4 things in this talk. First, a few funny stories. Then I want us to spend some time with my old friend. And then I'm going to tell you, I've never given this talk. I think we are confronting 2 major failures. I want to try to say what they are. They may be enormous opportunities. I'm modestly sure of what I'm saying. So all of this starts the day that I met Ilya and Mati. I think you've seen the picture. Ilya said, we're going to go have dinner with a bunch of my Finnish friends. And I says, well, great. Ilya says, you don't understand. So what's the problem? He said, they don't talk. I was not impressed. And we went to dinner, and there were 6 Finns and Ilya and me. And here was the conversation. Salt. Actually, it was a little longer than that. I think that was Mati. So another part of all of this is I wound up in Tampere as a Finland Distinguished Professors. Ilya and I don't know, did all of you see the movie long ago, Airplane? So Ilya and I were absolutely in love with Airplane. Among other things, there's the 2 black guys and they're talking jive, you may remember. And there's the purple-haired lady who explains, oh, I can talk jive. And we're in Tampere, and it's June 21st, and we're out taking saunas and running out nude on the long dock pier and diving into the cold water, explaining, I can talk jive, and just cracking up. So just lots of funny moments with this man I so love. Yesterday, I was flying home from London, and I want to tell you an experience that I had, and I hope that you will share it with me. We all know this guy. And I was partially asleep. Do you know what it is to be hypnagogic? The point where you're kind of drifting into sleep? I'm going to ask you to do this with me. It's not usual. First of all, let's picture the Ilya we all know and love with his brilliant blue eyes and this ready smile and this invitational welcome. Ilya welcomed. And of him, Lee said he was a superb scientist, but a better human being. What an amazing thing to have said about you. I realize how inadequate I am. I'm not an amazing human being. I'm an old Jewish guy. So please close your eyes. I'm going to ask you to do it. Picture Ilya and talking to him, and let yourself, I'm serious, become Ilya and see out through his eyes at the world Ilya saw, including you, with this sense of warmth and love and gentleness and openness and humor and fundamental sweetness. That's the Ilya we love. Not everybody has it. Ilya did, and Ilya still does. And Ilya Smolowicz, I love you. Thanks. I wish you were here, Ilya. It was very impolite of you to die. So I now want to talk about two failures. They both have to do with modeling. And Oli, thank you for leading us on this expedition. We think we know what we're talking about. There's a sense in which I think we don't. Yesterday, on the flight coming back from Houston, I was seated next to a brilliant young woman. She trained at MIT in material science, became a doctor, and she's a radiation oncologist associated with MD Anderson. And she's treating all kinds of cancers with specific agents for specific mutations in lung cancers and other kinds of cancers that induce remissions for a couple of years. And I said, well, do you know what an attractor is? She said, no. Well, I said, it's a model of a cell type as a pattern of gene expression. She said, oh. So she had no idea, and she's a well-trained oncologist. And here we are 50 years or 53 years after I happened to publish my own paper on cell types as a attractor, 52 years or 63 years after Jacob and Monod explained the mystery of why the same set of genes could express different sets of proteins by saying two genes repress one another. It's got two steady states, A on B off, B on A off. How come she doesn't know? Here is Sui, who has shown that cell types are high dimensional attractors. Here we are in this room thinking that routinely. And she doesn't know, and she's well trained and incredibly smart. Suggests that we are failing. Either it's the case that a concept such as an attractor has zero clinical relevance, or we haven't figured out what that relevance is, or we figured it out and haven't told anybody. Well, what's wrong with us? Is it really true that the idea of a cell type as an attractor has no relevance? And why aren't we finding those relevances and putting it into clinical practice? More broadly, there are those of us, I think I'm the first, almost maybe. So when I found out about Jacob and Monod in 63, I thought, there has to be this huge genetic regulatory network, thousands of genes. And I asked an odd question at 23. I asked, is there a class of networks that generically will exhibit ordered behavior? That question is the opposite of the normal question. The normal question is, gosh, what's this network and how did it evolve and how does it exactly work? And it's this specific network. And we know it in detail and can reach in and fiddle with it. I knew that I was doing it at the time, but watch the modeling move. Is there a class of systems that generically has a property? So I happened to invent random Boolean nets, and I happened to make two input nets because I thought that would be cool. And it turned out they were dynamically critical. And I thought, wow, order for free, because they have a small number of attractors and the lengths of state cycles are small and a lot of other magical properties. It took 30 years or 20 years to work out order, criticality, and chaos. I had no idea what that was. But it was obvious to me that this is some really powerful set of properties. Let me say some of cell types in a attractor. The different cell types are different attractors. Cell differentiation is a perturbation that moves you from one attractor into the basin of another. You can ask for any attractor, if you perturb things, how many other attractors can you get to? Well, you get to a few, and from them to others. So any cell type differentiates into only a few, and there are some others. These are generic properties of critical nets. 50 years later, we know that genetic regulatory nets really are critical. The work was done by Roberto Serra, separately by Sarah Walker. It's really true. Genetic networks are slightly subcritical. So we know these generic properties. Any cell type can differentiate into only a few, for example. Yet that's not part of clinical medicine, apparently. Why not? Why not? What are we failing to do? Now, some of us, including Lee and Sui and I, we happen to have formed a company. I thought years ago, I wrote a paper entitled Differentiation from Malignant to Benign Behavior, thinking maybe cancer cells are unused cell types, and you could trigger a cancer cell to being normal. And then Sui and I did some experiments years ago, and you can in some cases. There's worries about differentiation therapy. Sui will tell you herding cats. How come that's not a big part of cancer research? People aren't thinking that. Years ago, I'm trying to remember the name, there was a cancer biologist who had just confessed to failure in his reductionist approaches. This is around 11 years ago. He was at MIT, I think, and I called him and I said, could I come talk to you? He said, no, I'm a reductionist. I won't understand what you're talking about. Well, there's also that arrogance too. Meanwhile, we haven't succeeded, those of us who believe all of this. So I want to pose the first of two major questions. It's really true that genetic regulatory networks, for example, do have generic properties, but they have a hell of a lot of specific properties. We think as engineers, which is just fine, and if you're a molecular biologist, you surely do, and that's fine. Then we need to know the specific details of this system to perturb that touches specifically that. It'll put that, go do that, and then we'll cure the disease. And we have a whole bunch of things that treat very specific mutations in lung cancer, and it gives you a remission for a couple of years. Not bad. Where are we using the generic properties? And how are we using the thing? It seems we're not. So this is a failure that may be because you can't, or it may be a big opportunity. To pause over big opportunity, I think the fact that any cell type can change into only a few others is an enormous opportunity. And I want to tell you something that many of us know, but some of you may not. This is Harold Waddington in 1944. There's a mutation in Drosophila called bithorax, and it turns the metathorax into the thorax. So Drosophila has a second pair of wings. So it's called bithorax. And it's a major gene and part of a big complex. Waddington did some amazing experiments. He took a genetically normal fly. You can take the freshly laid egg and give it a little heat shock or a little ether shock. And some of the flies come out with a teeny bit of a transformation of their metathorax towards the thorax. You select them, and over cycles of generations, you keep perturbing with a little bit of ether or a little bit of heat shock. And over 10 or 15 generations, you pull out a population of flies that have the metathoracic phenotype. So did you pull out the bithorax gene? You did not. You pulled out 3 minor genes. Well, so it's polyclonal, it's polyfunctional. That means something fundamental. What did it? It means that managing to get the same phenotype can be achieved through alterations in a multiplicity of different genetic factors and the proteins that they make, right? But we're not using that in our thinking. And Wadd did that in the 1940s. So we're not using the idea. I mean, that idea is a cousin of the fact that any cell type can change into only a few others. It shows up in the metaplasias in transit termination and Drosophila, for example, in our gut metaplasia. And that's going to bring me to this other topic. But let's pick this. There's lots of generic properties. How do we turn them into clinically useful pathways of investigation and ultimately treatment? Sui and Lee and I are part of a group that we're cloning random genes that we will use to transfect cells and look at the phenotype induced by novel proteins in cells. What's lovely about that is it's an additive mutagenesis. We have no idea what these proteins will do, but we can damn well see. And damn well seeing what a protein, some funny protein does in a cell is not a computation on a digital twin. And that's the next point I want to turn to. Mathematics is spectacular. This is going to sound very weird. Let me tell you the Newtonian paradigm. Newton told me. Actually, he didn't, but here it is. Name the relevant variables, position and momentum. Write laws of motion coupling them, Newton's laws of motion, in differential form, ordinary or partial, stochastic. Define the boundary conditions, say of the billiard table. The boundary conditions give you all possible combinations of the values of the relevant variables. That's the phase space of the system. State your initial conditions. The billiard balls are here rolling this fast. Integrate the equations of motions, and you will get an entailed trajectory of the systems in its phase space. That's all of classical physics. It's all of quantum mechanics, too. Take the Schrodinger equation, state your boundary conditions, state your initial conditions, integrate, and you'll get an entailed trajectory of a probability distribution, and then whatever happens in measurement. So all of physics, all of classical sciences is in the Newtonian paradigm. So are our mathematical models. We assume we know the relevant variables. We assume, as Oli said, that we know the laws among them, and then we try to deduce the consequences. In our digital twins, we try to predict what's going to happen. I'm going to tell you that that's a big worry. So I'm actually going to recommend two papers that I'm the co-author on. It's with a wonderful guy named Andrea Rowley. He's a computer scientist. The first was published in Entropy in 2022. The title is "The World is Not a Theorem." The second took 3 years to publish. It was published a year and a quarter ago in the Journal of the Royal Society, April 14th, 2023, A Third Transition in Science. Please actually read it. Doesn't matter that it's Andrea. Let me tell you what we proved. We've proved you can use no mathematics based on set theory, which is all mathematics, to deduce the evolution of the biosphere. It's a massive third transition. Let me tell you briefly why. So I need some concepts. In 1790, Immanuel Kant said, an organized being has the property that the parts exist for and by means of the whole. I'm going to call that a Kantian whole. He didn't. You're a Kantian whole. You exist because you got a liver and a kidney and a spleen. They exist because they're part of you, right? All living organisms are Kantian wholes. We are. You'd have thought that people would now really understand that. We don't. For example, Richard Dawkins hasn't the faint idea about it. He thinks the organism is a vehicle. He's just wrong. Now, here are some steps. Given a Kantian whole, you can define the function of a part. Your heart does lots of things. It pumps blood. It jiggles water in your pericardial sac. It makes heart sounds. The function of your heart is pumping blood, not making heart sounds. Why? Because pumping blood is that subset of the causal properties of your heart that sustains the whole. Everybody got it? So the function of a part is a subset of the causal properties of a part that sustains the whole. But that very sentence means something astonishing. The same part has myriad causal consequences, any one of which might turn out to be a novel function. Well, Darwin thought of that. They're called Darwinian preadaptations or exaptations. Let's pause and go through this, because if you don't know about it and then know about it, it's going to change how you think. Dinosaurs had scales for thermoregulations. They got co-opted to flight feathers, totally different function. You have proteins that are lens proteins that are translucent or transparent. They came from normally colored enzymes. And the last example is my favorite. Some fish have a swim bladder. It's a sac partially filled with air and water. The density of air and water determines neutral buoyancy in the water column. The thought is the swim bladder evolved because some lungfish that splash around and can breathe got water in the swim bladder, and it was supposed to become a swim bladder. A new function arose in the biosphere, neutral buoyancy in the water column, and a new use was made of the same object, the lung. By partially filling it with water, it could detect neutral buoyancy. This means the following. I've got to give you one more sentence or two more sentences. Evolving biosphere keeps constructing novel uses of things. And the next thing I'm going to show you is we can't deduce the new uses. So this is sort of going to be the last step. This is going to mean that biological evolution keeps creating new relevant variables that are new relevant phase spaces, and we cannot deduce the new relevant phase spaces. So let me show you this part. Andre and I decided we really liked engine blocks, so everyone knows what an engine block is. Well, I'm talking about uses of things. That's not in physics, it's in biology. The function of a part is its use. What can you do with an engine block? Well, you drill 8 holes and make an engine. Andre is Italian. He says, you make 8 holes and you can store wine in it. He's right. Then I realized years ago, it's a rigid structure. You can use it as a chassis for a tractor. That's actually how tractors are made. And I will tell you the final 2 triumphs. It took about a year to get to this. The engine block is pretty heavy. It's a great paperweight, right? And then finally, in a great leap of imagination, the engine block's corners are rigid. You could crack open a coconut on the corners of an engine block. You can see why I was proud. Now, if you're using the engine block as a paperweight, can you deduce, I can use these things to crack open coconuts? You can't deduce it, right? I mean, it could have been a piece of banana. And even if you're using the engine block as a paperweight, you cannot deduce you can use it to crack open a coconut. So this means something absolutely fundamental. What's happening in evolution is that evolution finds new uses for the same protein. A protein can absorb a photon or conduct an electron or catalyze a reaction or bind a ligand or carry a tension mode or a compression mode. But you can't deduce the new use. That means that in evolution, molecules are coming to have novel functions that changes the phase space of evolution in a way that cannot be deduced. And that means we can't use any mathematics based on set theory proof. You cannot list all the uses of an engine block. You cannot list all the uses of a screwdriver. First axiom of set theory is 2 sets are identical if and only if they have the same members. But if you can't prove that the use of an engine block is identical to the use of screwdrivers, you can't use set theory. That means we can't use all the mathematics we know to deduce the evolution of the biosphere. This third transition is huge. And it's interesting that there's some physicists who are being approached about it. They won't go on the record to talk about it. So maybe we're wrong, or maybe they don't want to see it. So that's certainly true over a long time scale. Let's try to put it together with the first failure that I talked about generic properties. If some molecule comes to have a new use, you can bet that the generic properties of the system are going to be pretty much the same. So can we use generic properties when the engineering approach won't work, or we don't even need it? For example, can we find by phenotypic screening of some proteins molecules that do what we want and not try to deduce it? For example, Lee and Sui and I are interested in intrinsically disordered proteins. Well, we don't know how they fold and how they fold collaboratively with one another. Maybe you can use engineering when you have a molecule that has a well-defined binding pocket and either binds or doesn't bind, as in the old days with with allosteric enzyme. A vast set of our proteins are intrinsically disordered. My bet is intrinsically disordered proteins are really good at squidgling together to find new uses and new causal consequences that can't be deduced. So, okay, let's imagine that happens over dozens of years or 1,000 years or 1,000,000 years, or in a fungal bacterial community in the soil. Why do we think it doesn't happen in the lifetime of a patient who's accumulating mutations in a cancer? We've never thought about it. We've never thought about it. So what I want to end with, having babbled, and I think this is comprehensible, we have two big failures that may be enormous opportunities. We don't know how yet, we haven't paid attention how yet, to use the generic property of the system to find new diseases and disease treatments. Either it's impossible or we just haven't thought. And we certainly haven't told anybody about it. Meanwhile, in the lifetime of a patient with COVID, how do we know that some mutation isn't happening where a molecule folds in a slightly different way? And it's not that it no longer catalyzes a reaction, maybe it's carrying a structural load and that's how it works. We haven't the faintest idea. We haven't looked for it. We're thinking like engineers of the little yes-no logic in the Boolean function where we know what they are. They're little logic functions. They're not, they're molecules. So this seems to me to be harrowing and kind of wonderful. It's mathematics and it's somehow beyond it. It's biology. I don't know what it invites us to do, but it seems to me that it's pretty big. So with all of that, I'm going to stop again and tell you, Ilya, how much I love you. And if anybody were here who would tell me this is just full of crap, it'd be you. Thanks.